Hello everyone, welcome again. Today in this video I am going to talk about differentiation point between gram positive and gram negative bacteria. So basically gram positive and gram negative bacteria are differentiated on the basis of their cell wall structure, and after gram staining gram positive bacteria retain the stain and appear purplish or dark violet in color under microscope, but gram negative bacteria do not retain the stain. That's why they appear pinkish or reddish in color after the counter stain safranin. So this is the first main difference here. uh, Here it is right that gram positive bacteria appear as dark violet or purple color under microscope after gram staining, but gram negative bacteria appear pink or red colored after gram staining because they are stained with counter stain, that is safranin, and safranin are rejected by gram positive bacteria, but gram negative bacteria retain the safranin color, that is pink or reddish in color. And here you can see gram positive bacteria. In the gram positive bacteria there are two membrane, one is peptidoglycan cell wall and next one is plasma membrane, but in the gram negative bacteria there are three cell layer. Last one is, that is outer membrane, which is very thick and made up of lipopolysaccharide. Inside the outer membrane there is another membrane that is made up of peptidoglycan, or inside the peptidoglycan there is a plasma membrane. Here you can see the difference in between plasma membrane and peptidoglycan cell wall. There is a minor space that is known as periplasmic space, or which is very small in gram positive bacteria, but in the gram negative bacteria there are more periplasmic space between the outer membrane and the peptidoglycan layer or peptidoglycan layer and between the plasma membrane. So basically there are two times periplasmic space, which is very thick in gram negative bacteria, and in the gram positive bacteria this is very less in amount. Periplasmic space is almost nil in gram positive bacteria. So this makes the difference between gram positive and gram negative bacteria. Here in the gram positive bacteria cell wall is a single layered. You can see that cell wall is a single layered and it is a straight or even. Straight and even means it is not a wavy in a structure, but in the gram negative bacteria cell wall is a two layered and uneven, that is wavy. And here we can say that gram positive bacteria, in the gram positive bacteria S layer is present, which is more near towards the peptidoglycan layer, but in the gram negative bacteria S layer is present near the outer membrane. So in the positive S layer is toward peptidoglycan wall, and in the gram negative S layer is toward outer membrane. Here cell wall is very rigid and less elastic, but in the gram negative bacteria cell wall is less rigid and more elastic, because here lipid content is present outside on the gram negative bacteria. In the gram positive bacteria high peptidoglycan content is present in cell wall, and in gram negative bacteria less peptidoglycan is present in the cell wall. There is a acid which is muramic acid. Muramic acid content is more in gram positive bacteria, that is 16 to 20% of dry weight, but in the gram negative bacteria muramic acid content is less in cell wall, that is 2 to 5%. And if talk about the susceptibility or resistance, so we can say gram positive bacteria is highly resistant to the physical disruption, but gram negative bacteria are less resistant to the physical disruption, or we can say more susceptible to the physical disruption. Another acid, which is teichoic acid, is present in the gram positive bacteria, but in the gram negative bacteria teichoic acid is almost absent. As I told earlier that periplasmic space is less in the gram positive bacteria, but periplasmic space is more in gram negative bacteria. Here outer membrane, which is made up of lipopolysaccharide, is absent in the gram positive bacteria. That's why they do not retain this. That's why they retain the stain and do not wash away the stain. But in gram negative bacteria outer membrane is present, which is made up of lipopolysaccharide. That's why all the content of staining soluble inside the lipopolysaccharide and which washes away after the staining with alcohol. That's why outer membrane is present in gram negative bacteria, which makes it gram negative, because it do not retain stain due to this outer membrane. In gram positive bacteria lipid content is very low, but in the gram negative bacteria lipid content is very high. And lipopolysaccharides, which is a membrane, usually absent in the gram positive bacteria, which is present in the gram negative bacteria. Mesosome, which help in the respiration, is more prominent in the gram positive bacteria. These are basically the enfolding of the cell membrane, and Mesosomes are less prominent in the gram-negative bacteria. In the gram-positive bacteria, there is a flagella which help in the movement or locomotion with the 2 rings in basal body. But here, in gram-negative bacteria, flagella with 4 rings in basal body. Here only L and P rings are present, but here L, P, S, M, all 4 rings are present in gram-negative bacteria. Here, gram-positive bacteria usually produces exotoxin, but they produces usually endotoxins, and they are more susceptible to antibiotics. Gram-positive bacteria are more susceptible to antibiotic, means they are less resistant to antibiotic, but gram-negative bacteria are more resistant to antibiotic. They show high susceptibility to degradation by lysozyme enzyme, which degrade the enzyme of the cell, and here cell wall is less susceptible to degradation by lysozyme enzyme. Here porins are present in gram-positive bacteria, and in gram-negative bacteria porins are in gram-positive bacteria porins are absent, and in gram-negative bacteria porins are present. Porins are basically help in the movement of minerals or movement of ions from one side to another side. Basically they help in transportation. Here we can see example of gram-positive bacteria are Bacillus subtilis, Streptococcus, Lactobacillus, Clostridium. Basically rod shaped bacteria are included in the gram-positive bacteria, and in the gram-negative bacteria these are E. coli, Rhizobium, Vibrio, Acidobacter. So it was all about the differentiation or differences between gram positive and gram negative bacteria. I hope you like the video. Thank you, friends.